Welcome back, everyone. We heard two great, fantastic, how many adjectives are out there, social drivers and the clinical aspects of Alzheimer's. And now we will continue. We will move on to the second half of our program, where we are going to shift the focus to go deeper into biology of Alzheimer's disease and related dementias, exploring when we can learn from diverse model systems and from the genetics and lipid fat biology of the brain, and how these insights might guide the development of new therapies. Just a quick reminder, everyone is welcome to submit their questions on Slido. And of course, everyone at home, please do, as I've seen many questions coming in, continue to ask questions on the Slido link, which is behind me here on the board, as well as in the Zoom chat. So it is my pleasure to now introduce our next two speakers. Cara Croft, a senior lecturer in neuroscience and an Alzheimer's Society Dementia Research Leader Fellow at the Blizzard Institute, Barts, and the London School of Medicine and Dentistry, Queen Mary University of London. Dr. Croft's work uses innovative model systems, from mice to naked mole rats, to probe the mechanisms that drive neurodegeneration and to understand why some species appear remarkably resistant to age-related brain changes. Her talk, From Mice and Naked Mole Rats in Humans in Search of Therapies for Alzheimer's Disease, will take us inside this comparative approach and explore how insights from these models can inform the development of new treatments for humans. Following Dr. Croft, we will hear from Leyla Akay. She is the Director of Biology at Neurodegeneration NewCo, a startup. Dr. Akay's research focuses on how lipids and genetic variation shape brain health and disease, and how we can leverage that knowledge to design more precise therapeutic strategies. Her talk, Lipids in the Brain, What Genetics Can Tell Us About Developing Therapies for Alzheimer's Disease, will highlight how advances in human genetics and lipid biology are converging to reveal new targets and new hope for interventions. As with our first session, after each presenter, I will engage in a very brief conversation, but then I promise you, at the very end, all the presenters will join me on stage, where we will have a Q&A, both with your questions and the questions that are coming in from online. And with that, I will now be delighted to introduce Cara Croft.
Hi everyone, thank you for the invitation. I'm delighted to be here, and hopefully in the next 20 minutes I can give you an introduction to some of the exciting things that we're doing in my research group based at Queen Mary University of London. So just a quick insight to the title. We are using a comparative neurobiology approach with the hope that using all of these different models, being able to use these models with the correct questions, we can actually hopefully bring treatments to people living with Alzheimer's disease. So of course we have the mouse, who is a workhorse in biological research. We have the naked mole rats, which you might have heard about more from cancer and pain research, but hopefully I'll introduce you to why I think they're going to be really interesting in dementia research. And then of course humans, which we've already seen some of the clinical aspects and side of studying humans, but also how we can study humans in a dish in a laboratory, and that's what we're doing in my group as well. So the Alzheimer's disease brain in a nutshell. In a healthy brain, we have all these healthy brain cells with functioning connections. And when this goes wrong in Alzheimer's disease, these connections break away, and that might be why some of our loved ones have symptoms such as memory loss, cognitive decline. Now, we think some of the culprits are the amyloid beta plaques, as well as these neurofibrillary tau tangles. So today I'm going to focus on these neurofibrillary tau tangles, and you can see here the green stained tangles are what you would see under a microscope in a human brain, and that's what we're trying to look at in a lab as well. So many of you will be aware that these diseases are occurring for a long, long time before someone presents in a clinic. So not just Alzheimer's disease, lots of other neurodegenerative diseases as well. And 20 to 30 years before someone might present in a clinic, they have this amyloid plaque build up as well as neurofibrillary tau tangles. And this is one of the biggest battles, that these changes are building up in the brain way before anyone knows that that's happening. In response to this, we think that other brain cells kind of come in, try and help, try and respond to this, become on high alert to respond and hopefully clear this away. But what actually happens is that eventually triggers these brain cells to die, and then the memories fade away with that neuronal loss. And I've added these arrows in because I think this is quite a big cycle, initiating and self-initiating, but we still don't really know how to kind of approach this. Before then, in the clinic, someone appears with the memory loss, with other heterogeneous symptoms of Alzheimer's disease or other dementias, and on a classical MRI scan, the loss of brain cells. So one thing I would agree with the field is that targeting these diseases as early as possible in this biochemical phase is going to be the most important and probably where we have the most impact. So I've already mentioned tau in these tangles, and this is going to be a focus of my talk. And I think it's one of the most interesting proteins in dementia research. But when things are interesting, they're also really complicated. And I'm just going to introduce to 4 key angles of why it is so complicated, but also so interesting to study it in a lab, and then also bring you into the opportunities and challenges that it presents. So Jennifer kindly introduced us to a PET scan, and you can see here in Alzheimer's disease, this is tau pathology in the brain, but you also see tau across all of these other diseases as well. So that makes it really complicated because these other diseases have different symptoms, but most of them do share some signs of dementia as well. Adding to the complicatedness, then you get tau clumps and tau protein buildup in lots of different cell types. So we focus on the neurons, the nerve cells of the brain in Alzheimer's disease, but in lots of these other dementias, the protein tau builds up in lots of different cell types. So that also adds to the complicated nature of studying this disease. Now, when a gene produces protein, there can be lots of different proteins formed, and this molecular heterogeneity is something that we definitely get with tau. So you can see here 6 different isoforms that we have in the human brain. There's also one found in our periphery as well, and there's lots of others in all these different mammals. So that also adds to the complexity of studying this disease. And then lastly, when we come to structure, proteins normally fold in specific patterns and ways, whereas in these different dementias, they actually fold in different ways. So that also adds to the complexity because we might need to unpick different structures and pull them apart for all of these different diseases. But that makes it really exciting. So tau as a target, for Alzheimer's disease and moving into the clinic, there are actually several drugs already in clinical trials looking at different ways to change this tau protein and potentially as new therapies for people living with Alzheimer's disease or living with these other dementias. Now, to introduce you to my world, I'd like to kind of set the scene with tau and why it brings some challenges, but also why I think actually it brings more opportunities and why it's my area of passion and why I'm working on this disease. So definitely in terms of the challenges, when tau clumps up, lots of different changes happen to the tau protein, and that adds to the challenge because we need to understand which form or which species of tau do we actually need to target. And if we target the wrong one, will that actually affect the progression of disease, and will someone still have the memory decline that we don't want them to have? Tau as well actually really helps us in our brain cells. So it's there for the structure, it allows our cells to talk. So because there are these physiological roles, we don't want to just get rid of it completely. And I think that is one of the challenges when we are going after tau as a target. Now, sat in the middle, I think these are challenges, but also opportunities. So there is this relationship between taus and tangles, but we know the more tau tangles there are, the more neurons get lost. So we know there that it is important when it comes to brain cell health and brain cell loss. And then similarly, we know the more tau tangle burden there is, then there is the loss of cognition and cognitive decline in Alzheimer's disease. Now, an area that has been a challenge has been how to study these tau tangles in a lab. And I'm going to take you through some of the ways we're doing that in my group, but I'd say in the last decade, there's been some really innovative ways that we can now study tau in a lab, which does make it a really exciting drug target. And then definitely on the opportunities side. So the genetic causes of Alzheimer's disease, there's certain genetic factors which will predispose less than 1% of the people to 1% of the population to get Alzheimer's disease, to definitely get Alzheimer's disease. But some of these individuals escape that, and that is seen in the restriction of tau in their brains. So because these people are resistant to this tau pathology, we think this is why they've managed to avoid Alzheimer's disease or other dementias. And then also, as Jennifer alluded to, now that we can measure tau in a simple blood test, we can probably test whether the tau medicines going into the clinic are doing a good job. So I think that really does bring us a lot of opportunity. So moving on to work in my group, as I've mentioned, models to study tau pathology in general have been quite mixed. There's sex differences in animals with tau pathology that we've induced to have tau pathology. You can't really just you can't really just model it in cells. So what we've done is a way where you can keep small sections of mouse brain tissue alive. We use viral induction to change the genetic makeup of these sections. And what you can see, hopefully, is that we're able to model those Alzheimer's disease tangles very similar to what you do actually see in that Alzheimer's disease brain. But this is a really nice model because it's easy to manipulate. We can treat them in a dish in a space of 2 to 4 weeks. Previous models, you'd typically be waiting 12 months to see this pathology. So that's really important because we want faster approaches to be able to study tau tangles. And then our colleagues have also used the same viral induction, and you get less of the heterogeneity, but you also get tangles in living mice, also that then develop cognitive decline by injecting these viruses as well. And that's in 3 months. So now we have these mouse models that we can study tau pathology. So one thing to note is that these are progressive neurodegenerative disorders. And what I saw when we were looking down the microscope at these tau tangles in a dish is, yes, they do develop rapidly, but also what you'll see in that bottom corner of that panel is that they do develop progressively, but they're also stable for a long time. And in other work, we can see these tangles there living in neurons in these sections in a dish for 3 months and beyond in culture. So that gives us an opportunity to start looking at some of the earliest events associated with these tau tangles. But one key thing that I did note when we were looking at them under the microscope is the fact that they're there for this very long time and they don't just suddenly cause a brain cell to die. And I think that's really important because I think the thinking in the field was that tau will automatically kill a cell. But this gave us the idea that there are potentially compensation mechanisms that we should be looking into. So how do you start to look at the environment of a tangle? How do we look at them? So one way we looked at this was there's this fluorescent protein, so a glowing protein that we can then induce to change colour. And when you induce this colour change, you can then track it for time afterwards. So then you know that you've shone a light on it, and that you've got your old protein there that you'd seen a week ago or 2 weeks ago. So by changing it from green to red, we can actually follow it over time. And I think this is really important because this allowed us to follow individual tau tangles for a long time. So just one thing to point out is that a lot of work in the Alzheimer's field or in dementia research just focuses on one particular snapshot, whereas we wanted to really use the advantages of preclinical models to be able to look at some of these earliest events, look at them mid-stage, look at them longer. And that's what we're trying to do by looking at things in 4 dimensions in my lab. And by 4 dimensions, I mean 3D space. So we have these 3D cultures, and then time, looking at early stages and late stages. So by using this approach, we can now track new and old tangles over time. So you can see here one of the earliest tangles that form, we shine a light on it, and you can see that now there's these 2 populations of protein. But what you can see over time is that this red starts to decline. So that's showing us that the tau over time is actually unraveling. So the brain cells themselves have evolved mechanisms to be able to deal with this pathological insult. Now, we thought this was really exciting because the cells aren't just dying. And actually, maybe this is a process that we can actually boost using medicine, so helping brain cells help themselves. And that's kind of where I'll come back to towards the end of my talk, where we think we can now use drugs to potentially boost brain cell clearance of tau. So one thing to note is that we did this when tangles were really new. If you do this photo conversion and follow the tangles after an extended period of time, what you actually see is that when a tangle's been around for longer, the clearance is much slower. So this really means that any therapies need to go in right at the start when tangles are newly formed, when we think brain cells are able to kind of help with this clearance and help with this waste disposal much sooner. So moving on from mice to naked mole rats. So if you've not come across these guys before, they're funny. Everyone always laughs when they see them. And what's really interesting is that they live for such a long time. So they are a natural superager, and that's why we're interested in them. Because based on their size, they should probably only live for 2 to 3 years. But they actually live for 35 years or more. And I've been so lucky to have access to them at Queen Mary University of London and also to be working with excellent collaborators. Now, when it comes to dementia, we're really interested in them because they don't appear to get amyloid plaques and they don't appear to get tau tangles. And that's essentially what we think we need as humans to have long-term brain health. One thing that has been reported by colleagues in the field is that their tau molecular structure actually appears to be longer. And that's something that I'll come back to, but there are also similarities to our tau protein as well. So we want to explore whether there's resilience or resistance to tau pathology in the naked mole rat. So one thing that we did find was that during aging and as these naked mole rats get older, their tau appears to get longer. Now, we're still exploring why that is, but what we think is that that actually helps keep the neuronal structure more intact and more integral for longer periods of time. So if there is a strategy that we can do that in our own brains, then that might be something that will help keep brain cells healthier for longer and not have tau clumps. Now, the other factor that we've been exploring and we've been able to reproduce as well is that actually tau phosphorylation is something that happens when tau starts tangling and clumping up in the brain, whereas naked mole rats have a lot of this at the start of their lifespan, but reduce it over age. So we want to understand how they're reducing that, and maybe that's something we want our brain cells to do. And then lastly, we wanted to manipulate this more. So we do a lot of manipulation within my group and introducing dementia-linked mutations in order to study things. Like we've shown already that we're able to study the tau tangles by introducing mutations with viruses. Then we were able to introduce some dementia-linked mutations to make naked mole rat tau. And what you actually see is when we do this just with human tau and add these mutations, then you get a lot of buildup of the green neurofibrillary tangle tau protein. Whereas when we do this with naked mole rat tau, there is very limited accumulation of these tangles. So this is something, again, that we're still exploring, but we think because it's longer, it's able to resist these changes, which within human tau structures actually causes them to accumulate and clump. So lastly, as I've mentioned, studying humans is really, really hard. 20 to 30 years, these proteins and tau clumps are building up, and we need to be able to study this faster in a lab. So lots of other labs have pioneered the use of these human brain organoids, typically starting off as skin cells or even blood cells, and you can convert them into brain cells and bundles of brain cells in these organoids. Now, what we've done is we slice them really thin, and that allows us to keep them in culture for a long time. And when we add fluorescent markers, we can actually see the structures of these neurons and be able to visualize and grow them for a long time. Now, this is very much fresh work that we've kind of been working on for the last year, but we're now able to see these tau clumps and tau tangles in human organoid brain slices. So we really think this is an exciting way that we're moving from mouse to human models, but also potentially reproducing what we also see in mice and kind of highlighting the use of all these different model systems, being able to compare them to understand more about our brain health. And using this approach, we also want to kind of look at the tau clearance and the mechanisms of this tau clumping, but within these human neurons in a dish. Now, lastly, I highlighted that boosting tau clearance could potentially be a strategy. We do want to help brain cells help themselves, and that's something that we're working on. So there are several key mechanisms that we know that proteins can unravel and unclump, and that's something that we want to kind of boost. So first, we're going to look at the key mechanisms of how neurons are able to unravel this tau pathology, and then work with the Alzheimer's Research UK Albarada Drug Discovery Institute to test the capacity of tau clearing therapeutics in these human models, with the idea that some neurons already have this fast tau unraveling, and we want to normalize them all to have this really fast tau unraveling, with the idea that this could be future treatments for Alzheimer's disease or dementia. So just to wrap up and leave you with some take-home messages. So modelling tau and neurofibrillary tangles is no longer a bottleneck. There's lots of exciting models being used in labs around the world that make it faster to develop tau therapeutics. And hopefully I've shown you how we're using mice, naked mole rats and human brain slices now to start looking at these interesting tau clumps. What we've actually shown is that brain cells have evolved these mechanisms to unravel tau, but it will be really important to boost this unraveling much sooner. Naked mole rats are a superager. They're a model of longevity, and we really think they're providing a unique insight to our work as well. And then lastly, that we think that enhancing tau clearance rates may provide novel therapeutic avenues. And our hope is that we ultimately are able to determine the causes and consequences of these tau clumps and tau inclusions, and how best to slow, prevent or cure tauopathies like Alzheimer's disease. And with that, I'll thank my lab and my funders, which have believed in some of these crazy ideas, but hopefully will actually be beneficial to humans. And thank you for your attention.
Thank you very much. And the naked mole rat, it has captured the imagination, not just the scientists, but the public. Yes, I did a little deep dive, and Disney does have a plush toy of a naked mole rat, Rufus. And so my question, of course, will be related to the naked mole rat. That was fantastic. And really to underscore the importance of basic biology, to let us understand ways in which we can intervene in these processes. So my question is, the difference between the naked mole rat and human tau seems interesting and could be critical, right, to their healthy brain aging and not getting these tangles. Do you think there are other features of their brains, neurons, that might help?
Yeah, so unfortunately in 20 minutes, I'm not able to fit in everything that we're doing with these interesting characters. But one line that is really interesting that we're pursuing is the fact that we can actually keep naked mole rat neurons alive in a dish as well. And we've challenged them with Alzheimer's disease tau, and it appears that they have mechanisms where it doesn't clump up. So we think if we can make our human brain cells like that, then that might be a way that could be a therapy in future. And I think that's really exciting. And I'm excited to get back to London to see what the latest findings are as well.
Do you miss your little critters? Yeah. They are the model for studying longevity across the board, not just aging, the Alzheimer's, aging-related diseases, but also longevity in general. But thank you very much, Cara. We will have you up here at the very end.
Thank you.
It is now my pleasure to introduce Dr. Leyla Akay. Thank you.
Hi, everyone. I'm so excited to be here, so thank you so much for the invitation to speak tonight. I feel like I already learned so much about Alzheimer's disease, so my brain feels all nice and fluffy. My name is Leyla. I am a neuroscientist by training, and currently I am the director of biology at a very new startup company here in Boston, and it's focused on Alzheimer's disease. So I'm excited to share a little bit about what we've been doing just down the street. I wanted to kind of add a little historical perspective to how we think about Alzheimer's disease, starting back in the early 1900s with Dr. Alzheimer himself. So Dr. Alzheimer was a German psychiatrist. And at this time, the field of psychiatry was really emerging, and it wasn't really clear to us where psychiatric illnesses came from. And so for doctors like Alzheimer, who believed that there were biological causes to these psychiatric illnesses, there was a tremendous amount of pressure on him to prove that. So the way that they would do this is follow interesting case studies of patients with different kinds of psychiatric illnesses and kind of meticulously document all of their clinical symptoms. And then after they passed away, they would do a post-mortem brain autopsy. And the pressure was on to try and find something in their brains that could be used to link this to whatever kind of psychiatric symptoms they had. So under this kind of debate that was going on, Dr. Alzheimer met this patient, Auguste Dieter. And Auguste was brought to his hospital by her husband. She was only 50 years old, but she had experienced this really rapid onset of paranoia, memory loss. It was so severe, she was lost within her own house. She would, start sentences and then kind of forget what she was saying in the middle of a sentence. And so Alzheimer agreed to take her on as a patient, and he followed her clinical progression over 4 years before she passed away from an infection. And then after she passed away, Alzheimer embarked on this post-mortem autopsy. And his goal was just to find and document everything about her brain that he thought could be different. So he wrote this case study about, the vasculature in her brain, how it was twisted and tangled, the fibers he could find inside neurons, what we now call neurofibrillary tau tangles. And he noticed that there was a special substance in the cortex, these plaques of sticky proteins that marked her brain. So he presented his findings at a conference, and he didn't get any questions, which was very sad at the time. But now we know that this disease is actually incredibly important and very, very prevalent, unfortunately. So that special substance of sticky plaques that he first described in her brain are what we now know to be amyloid beta plaques. And, since that kind of initial discovery, we've been really focused on chasing these amyloid beta plaques and trying to develop ways to remove them. And I just wanted to introduce, we heard a little bit about donanemab, which is one of the kind of FDA-approved therapies that reduces brain amyloid. So this is a monoclonal antibody, and as you can see from this graph here, it really, really effectively gets rid of brain amyloid. So in orange, those are people who are on the drug, and you can see over many weeks, the amount of amyloid in their brain really reduces. But unfortunately, these patients still experience cognitive decline. And of course, it is statistically slower, but ultimately these patients are declining. And I think that it's fair to ask if we can do better, and if we can say, are there other potential, causes or biological mechanisms contributing to Alzheimer's disease. And I think a really good way to do this is to kind of turn to this question of human genetics and to ask, within the greater population, which genes predispose us to having an increased risk for Alzheimer's disease. And by far, one of the greatest risk factors genetically for Alzheimer's is a gene called ApoE4. So ApoE4 is common in about 15% of the general population. So about 1 in 7 people have a copy of this gene. But in Alzheimer's disease patients, it's about 40 to 50% have a copy. So it's really strongly enriched. Having 1 copy will increase your risk about 4 to 5 times, but having 2 copies increases your risk 8 to 12 times. So this is a major genetic risk factor, and it is incredibly common in the population. What does ApoE do? Well, the ApoE gene codes for a lipid transporter. So this is a protein that specializes and binds to lipids and kind of transports them in the brain. And it moves the lipids or the fat molecules from cells that need them to the cells that take them up. And you can see here, I've just kind of sketched out, this is the ApoE3 gene. So this is kind of the neutral version of ApoE. And ApoE4 is just different by 1 single amino acid. So it's a very small change. But we think this has profound structural and functional consequences. So the ApoE3 protein is able to kind of structurally form a lipid particle that can bind and transport lipids, but ApoE4 is less structurally stable, and because of this altered kind of 3-dimensional structure, it binds fewer lipids, and the lipoprotein particles are much smaller. So ApoE4 is just worse at transporting lipids in the brain. But how does that increase risk of Alzheimer's? What does that have anything to do with Alzheimer's disease? So the way that we tried to tackle this question was first by performing something called RNA sequencing. And so here we took the brains from individuals, donors, who either had ApoE3, which is the common healthy version, or ApoE4, which is the risk version. And we're able to isolate individual cells from these brain tissues and then look inside the cells and ask which genes are being turned on and which genes are being turned off. And this gives us a little bit of insight into what biological pathways are now being changed by this risk genes expression. And so what we found is that there was massive changes to lipid metabolism-related genes in these ApoE4 carriers. And I'll just kind of divert your attention to specifically cholesterol metabolism machinery. And we found that these genes were really increased specifically in oligodendrocytes, and these are the cells that produce myelin, or the fatty insulation of our electrical wiring in the brain. Next, we wanted to see what that actually looked like inside the brains, so we performed some immunohistochemistry, which is staining, and we could see that in the brains of people with the ApoE3 gene, the cholesterol is nicely co-localizing with the myelin axon, so this is what we would expect, this is good. But in the ApoE4 individuals, the cholesterol is balling up. It's accumulating inside these brain cells. Since the oligodendrocytes are responsible for generating myelin, we then wanted to see whether their myelin would be any different. And indeed, we can see that in the brains of the ApoE4 carriers, there's really a disruption to this electrical insulation. So you can see here with this myelin staining, the kind of macrostructural pattern is really disrupted in ApoE4 carriers. And these gray images here on the right show that there's, even at the electrical level, there's a difference in the myelination. So at this point, we've kind of described this phenomenology that ApoE4 is linked to cholesterol accumulation and myelin damages. And we wanted to understand if these two phenotypes are kind of linked, which we think they are. So what we did was we take this mouse model, and these are mice that are engineered to express the human ApoE4 protein. And what we did was we treated them with a drug that kind of helps move cholesterol in the brain. And we found that animals, the ApoE4 animals that were treated with this drug, have a much increased amount of myelination. You can see those dark, thick rings on the bottoms. That's like the cross-section of the myelin. And encouragingly, when we test these mice on a functional task, you can see that the mice that got this drug performed a lot better. So in orange, these are the mice that we give the drug to, and every day we test them on like a little memory task. And so they're able to do it quicker and quicker and quicker, which means that they can learn and remember things a little bit better. So just to kind of summarize that first point, we're really excited to think that ApoE4 may be this massive genetic risk factor. One mechanism could be through disruption of lipid metabolism inside brain cells. And I just want to take a quick moment to point out, though, that there are many different risk factors, risk genes related to Alzheimer's disease, and many of them actually also seem to be implicated in lipid metabolism. These could be receptors, for example, sortilin. This is a receptor that interacts with ApoE. These could be other lipid transport molecules, for example, ApoJ, or lipid transporters that help put lipids onto ApoE, for example, ABCA7. So all these many different risk factors, many of them are linked to lipid metabolism, and kind of crucially, these can lead to different downstream effects. These can lead to impairments in metabolism, these could lead to changes in inflammation, or just an accumulation of lipids in the wrong places in brain cells. Okay, so the next question is, well, what do we do with all of this, and is there a way that we can translate some of these ideas into hopefully therapies? So our next project was using something called CRISPR technology, or genome-wide CRISPR screening. And so what we did is we take our models of these ApoE4 brain cells, and these are cells that have been loaded up with all of these lipids, and we use CRISPR to delete every single gene in the human genome and then ask which genes kind of modify this lipid phenotype. So we do this for all 20,000 genes, and that's kind of what the data looks like, but I'll kind of break it down. So from this big data set, we're able to ask which genes are really significantly modifying this phenotype of lipid accumulation. And we pull out about 300 different genes that we think are super important, and we can ask, are any of these genes all falling into like a similar kind of biological pathway? And we find that there's this enrichment of a pathway called the Wnt signaling pathway. And this is a pathway that's like very highly studied in development and a little bit so as well in neurodegeneration and aging. But what kind of really piqued our interest is that the Wnt signaling cascade is modified by a really famous tau kinase called GSK3 beta. So we thought, oh, okay, interesting. So we took our cells, and these cells have lipid droplets. You can see them there in yellow. These are the lipid accumulations. And we treated our cells with a molecule that inhibits GSK3 beta. And we see a reduction in this lipid accumulation. We wanted to understand a little bit better how this was happening, so we ran some RNA sequencing, and we see that these cells start to express genes related to oxidative phosphorylation and autophagy. And so we can do some sort of imaging and track the mitochondria and the lipids in these cells before and after the drug treatment. And we see that after drug treatment, the lipids seem to be co-localizing with the mitochondria. So we think that they're kind of promoting the mitochondrial ability to use these lipids and hopefully generate energy. Next, we took a look at our mouse models. And so these are the brains of mice that are either expressing ApoE3 with a tau mutation or ApoE4 with a tau mutation. And yellow is the BODIP stain. So this is a lipid droplet stain. And you can see it is insane. In the ApoE3 mouse, there's some lipid droplets, a little bit, but in the ApoE4 mouse, there is a ton of lipid droplets. And these lipid droplets are really accumulating in almost every single major cell type. So we wanted to test our hypothesis that targeting this pathway could have some sort of beneficial effect. And so we administered this GSK3 beta to these mouse models, and we see nicely that there is a reduction in the lipid droplet accumulation, as well as an increase in the myelination. So given these effects, we were kind of really interested about taking this data and just running with it, since we don't think that anyone else is doing this right now. And so we decided to start a new company. It's called the ApoE Company, or TAC for short. And this is done in collaboration with Ole Isaacson. He's a professor here at Harvard Medical School, my PhD advisor, Li Wei Tsai, as well as Doug Trico, who's a kind of biotech veteran. And so we got together and decided to just take this idea and run with it. So our approach at TAC Therapeutics is basically asking, can we target Alzheimer's disease not by focusing on clearing amyloid beta from the brain, but from first and foremost targeting lipid accumulation. And so we're relying on using very unbiased target identification strategies. These could be genetic or chemical, really with a goal of asking, can we restore an ApoE4 function? Can we make ApoE4 act a little bit more like ApoE3 and hopefully correct these lipid defects? And I just wanted to give you a little kind of teaser of some of our data because I'm really excited about it. So like I showed you before, these are our ApoE3 cells, and now we're showing a different brain cell type called the astrocytes. And you can see with the kind of normal version of this gene, ApoE3, there's really minimal lipid accumulation shown in red. but with the ApoE4 cells, there's a lot of this lipid accumulation. And using one of the hits targets that we've identified, you can see that we're able to really reduce lipid accumulation in the ApoE4 cells. And kind of intriguingly, we find another hit that is able to kind of boost lipid accumulation in the ApoE4 cells and seems to really change where in the cell these lipids are, their kind of distribution. So we think that there's a lot of room for new kind of research. There's a lot that we don't know, but we're excited to kind of go forth. And then finally, I just wanted to kind of bring this back to this original concept of Alzheimer's, first case study, where he talked about all of the things in Auguste Dieter's brain. Because in addition, to the vasculature, to the amyloid beta and the tau tangles, he also mentioned that in her brain, the glial cells had accumulated fat sacs. And this is now what we think are lipid droplets. So I think, there's some historical precedent, hopefully, to be studying this kind of lipid accumulation, thinking about this as truly a pathology of Alzheimer's disease. Okay, and so with that, I would like to thank you and thank everyone here.
Thank you, Leyla. That was fantastic. So it's really the lipid. That was a remarkable photo. I mean, it was just really hard. I mean, there's no real science there. It was just very remarkable. I had a brief question before we open it up to the audience and the in-person and the online audience, whether there are protective forms of ApoE, and what do they tell us about the disease?
Yeah, that is such an important point. So we talked a lot about this major risk factor, ApoE4, but the truth is there's so many mutations in ApoE, and one of them, called ApoE Christchurch, is incredibly rare, but it's so protective. And as our previous speakers kind of alluded to today, this mutation was found in a family that has this familial like Alzheimer's mutation, where they will get Alzheimer's at the age of 30. So it's an incredibly aggressive form of Alzheimer's disease, but one woman escaped this. So she did not have Alzheimer's, even though her brain was full of amyloid beta, and she was homozygous for this ultra rare mutation in ApoE called Christchurch. And this is a very recent discovery, so only kind of recently we started digging into like the biology of what this might be. But I want to point out that ApoE3 Christchurch has been shown to really preferentially extract damaged lipids from cells. So it seems to have this enhanced ability to take up lipids that are toxic or damaged and really help drive them. So I would like to argue maybe that is, part of the kind of protective effects of ApoE Christchurch.
So sort of like a trash removal system.
Yeah.
Yeah, wow. Well, that was fantastic. Wow, it's really exciting. And what's more exciting is that I'm going to bring everybody back up, and I'm going to showcase our brilliant speakers. I'm going to just sit here, and then, yeah, sure. And, well, I thought I would be asking all the questions, but I am not going to be so selfish, because so many questions have come in. And I want to give the audiences, both virtual and online, the opportunity to get their questions addressed by this panel of excellence. So I'm going to take the questions as they came in so that I'm not biased in any way. So this question could be for you, Leyla. How do elevated lipids, cholesterol, affect risk of Alzheimer's, and does taking statins for the elevations add to the risk of ALS?
That's such an important question. So people have really looked into this question of whether statin usage has any kind of protective effect on Alzheimer's. And the short answer is that it's really complicated and we don't have a good answer. I think part of that is that the kind of brain's pool of cholesterol is separate from the body's pool of cholesterol. So the cholesterol that's produced in the body and in the periphery is really not crossing into the brain. It's two kind of different separate angles. And of course, statins are lowering cholesterol by attacking the enzyme that makes cholesterol, and that might not be the problem in Alzheimer's. It might not be that we're making too much cholesterol. It could be instead that the cholesterol is not where it should be and is not being transported correctly. So I think I totally see where that question comes from. I think it's an amazing question. I just think it's a very nuanced.
Yeah, it makes sense because
Yeah.
it's used for, cholesterol control and lipids, so, right?
Yeah.
That's great. Okay, this question is for Ganga. Is there within group variation, RE, ADRD, if you control for SES, what would the causal influence share be, and what would be the mechanism?
So yes, one of the things that has persisted is that even when you account for differences in socioeconomic status, there are still clear racial differences where particularly Black Americans have higher rates of Alzheimer's and other dementias. And as I was kind of trying to hint at in my presentation, a lot of it, I think, has to do with the specific stressors that different groups are exposed to over the life course and the way that resources are distributed. So there is the stress piece of it, and then there is the resources that go along with that. So your ability to cope with stress and even access healthcare, which is a basic way of, maintaining our health, are going to be different between these groups. So there are complications to this that I hinted at. There are differences in the way that ApoE performs in these groups. And so there are definitely relationships between genetics and environment, but because we have strongly moved away from biological determinism, we do understand that genes are not primarily driving the differences in Alzheimer's risk between these groups. And so we sort of at this stage really propose that cause is attributable to different experiences of the social environment.
Great. So this question here, of course, is going to have to be you, Cara, because it has to do with the rat. Can you use human brain tissue from deceased donors as organ donor to study same effects as with the mole rat studies?
So we're focused on the naked mole rats as an extraordinary model of longevity, but there is the idea that some people avoid dementia all their lives and they can get to over 100 years old and there's no signs of dementia in their brains. So that's not something that's happening in my lab, but in other labs they are actually studying those brains and seeing what is the equivalent of the human naked mole rat. And I think some of the pathways may actually overlap, and that might be why their brains are kind of super cleaners, similar to like the naked mole rat.
It's interesting, and I'll get back to this. There was a recent New York Times article about the cognitive resilience of superagers. And so I'm not sure if you're familiar with that article. Would you like to comment on that and how that, yep.
Yeah. So in this study, they showed, they took these superagers, people with dementia, young people, and I guess people that had aged but hadn't got dementia. And what they actually showed, it's quite a controversial area, but there is a capacity for more new neurons being born in the brains of their superagers. So I think when this is extended to more people, understanding how these new neurons can be born may also be something that we might want to harness for therapeutics.
Yeah, it was an interesting article and a little bit controversial. This question is for Jennifer. Is there any evidence that the trace element lithium helps in humans?
Yeah, so this is a really intriguing question, and especially following the last talk that we just heard, because lithium is actually shown to be neuroprotective, or it's thought it's been postulated to be neuroprotective. And this is not just lithium that's used to treat bipolar disorder. But actually, lithium is a natural element found in our environment. So we all are exposed to micro amounts of lithium, and also through our diet. And so there's been mixed data on whether or not lithium can be neuroprotective. What's interesting is that lithium is a very potent inhibitor of GSK3 beta, which is an intersection with the lipid biology that Cara presented. So there was a recent publication from Bruce Shankner at HMS, which actually showed in a mouse model and also in human postmortem brain tissue that have all kind of metals and homeostasis, lithium was the one that was depleted in the brains of those with Alzheimer's disease compared to healthy controls. And they went on to carry out a series of experiments in mice where they depleted lithium. And actually this led to increased pathology and worse performance on tasks in the mice. And when they restored, and they found that lithium was actually being sequestered in the amyloid plaques. And when they replaced lithium in the diets, they actually were able to rescue this phenotype in mice. It was dependent on the formulations. Only lithium orotate, only that formulation of the salt was able to escape kind of being sequestered into plaques. So now at MGB, we're actually carrying out one of the first prospective clinical trials, starting with dosing of introducing micro amounts of lithium orotate into individuals with cognitive concerns and mild cognitive impairment, early dementia, to see if it can be safely tolerated. And then the next step would be, could it be efficacious? Could it actually be help prevent dementia?
And actually, there was a follow-up by this same person. What is your perception of Professor Bruce Yanker's lithium orotate exploration?
Yeah, I think it's incredibly promising, and and again, that was coupled not only with the elegant work in mice that his group carried out, but also again with evidence that links directly with David Bennett and the Rush aging cohort postmortem data, which is a very well characterized study of postmortem factors. So again, this was so it went from humans to mice, and also the experiments again in mice I felt were very rigorous. And there's also aside from that study, again, there's a larger question that's been out there, not only in psychiatry about lithium. Do patients on lithium, are they more neuroprotected? But there's been kind of this hint of this like in the literature for some time. So combined with the robustness of the study and kind of what we know about lithium already, and the fact that we're already getting exposed to it in our diets, I think it's incredibly compelling.
Very good. And this is for Ganga. Your focus is on external social factors affecting ADRD. What about internal familial factors, living with mental illness or physical abuse?
So that's a really great question. And I think I zoomed out on the social environment to look at social hierarchies as this sort of predominant environment, but the environment can be conceptualized as increasingly smaller from society all the way down to ourselves, right? And so throughout those in different levels of environment, you have similar systems of stress that operate. And certainly within the family dynamic itself, you see those stressors in place, and certainly differences in who is exposed to what type of familial dynamics is also a function of the larger social structures. And so while we can look at any sort of granular level of environment, it's helpful to always look at it in the context of the larger social environments that shaped those family dynamics, who gets mental illness, and how well they are able to treat it. And so yes, certainly those more internal dynamics within the family or within communities are definitely influential in those stress processes. They are themselves shaped by the larger social forces.
It's a very great response. So, Cara, again, without naming names, this is, so do you account for amyloid in your in vitro model and how amyloid buildup may influence the tau untangling?
So I think that's an important question. I think that is probably one of like the biggest questions in Alzheimer's disease, is essentially how amyloid talks to tau. And I think if we understand that, that is probably some of the earliest events. Within our modelling, we're focused on the tau purely, mainly because of that connection to brain cell loss. And we think that even if you potentially stop the amyloid pathology, the tau pathology will have already been triggered. And that is something that we do then need to stop as well in an Alzheimer's disease brain.
Okay. Leyla, this is caveated with probably a controversial question, but has anyone researched peptide therapy for Alzheimer's?
Molecules or
And that's the question, but we can move on. That's okay. What other effects outside the brain does the ApoE variant have?
Oh, I can answer that question. Peptides, I don't know. ApoE definitely it's associated with many different effects. predominantly like a lot of immune-related processes and inflammation. And in fact, there's this really amazing Nature Medicine article a couple years ago where they profiled the plasma proteomics of ApoE4 carriers. And regardless of disease, because ApoE is linked to many different diseases, the kind of consistent signature was this increase in immune and inflammatory-related protein signatures. So I think it's plausible to think that ApoE is also not only intimately linked to lipid metabolism, but like kind of brings lipid metabolism into very close communication with the immune system and can drive immune or inflammatory related processes.
Okay, Jennifer, please expand on PET research. How are you collecting data, and when are you doing, and what are you doing with it? Could you work with cancer patients and share data?
Yeah, so PET, this is a great question, and it kind of gets to the point that PET imaging is actually used clinically, because just without measuring specific molecules, we can measure overall brain activity, so-called like an FDG PET, which is a measure of metabolism. And this is often a very useful clinical tool in cancer patients to see if there's increased activity, which could be from a nidus of tumor cells, and to see how individuals are responding to treatment. So we are utilizing it. We do use just metabolic PET to kind of correlate that with areas that are shrunk in the brain, like are they also less active? And then also we focus more and more on PET scans where we can measure molecules together with metabolism. So we can measure forms of tau and amyloid and overlay that with functional changes. And we can measure this and see if it's a marker of how what will be their trajectory in years to come. So that's how we're utilizing PET imaging in our studies. And then in clinical trials, it's of course utilized to see if agents that we want to target amyloid or tau, if they're actually working effectively as well. So we do think that tau PET imaging may provide some additional diagnostic information beyond the blood test that we currently have for tau. So I think there will continue to be a utility, especially in the clinical research space, of tau PET tracers. And they're becoming more and more elegant, more and more specific and sensitive. And then the next level will be, can we measure other pathologies like TDP-43, which we know is also an important pathology in aging and cognitive decline. But right now we don't have a tracer that can measure in living individuals. So that will be kind of a next step in PET research.
Thank you. There's a question for you, Ganga, on the loneliness epidemic and its impact on dementia and Alzheimer's. Going up, going down? What do you?
Yeah, so in terms of understanding the complicated causes, right, we in general, as others have mentioned, have an aging population. And so because Alzheimer's disease and other dementias are so correlated with aging, it's important for us to be able to understand that is a large driver of the increase in prevalence that we're observing. But there are population level changes that are happening in our society that are also very influential, and not just Alzheimer's disease, but all of the other sort of chronic diseases that are impacted by things like social isolation. So we know as we've heard, that it influences not just our mental health, which then influences our risk for things like ADRD and cardiovascular disease, but also other health behaviors. And so if people are more socially isolated, they're less likely to engage in physical activity. And so there's this very interdependent cascade that influences health over the life course and in relation to Alzheimer's disease. And I think it's somewhat of a challenge, right, given the technological factors and many other changes in our societies that are sort of driving that. The more we know that, the more social media use, the more social isolation that people report. And so there are these ways that we understand that these changes are happening that at the population level that are influencing risk for disease, but it's very hard to sort of tease out exactly those mechanisms. But yes, I anticipate that part of the challenge in addressing an opportunity, as I like how Cara put that, for addressing Alzheimer's disease is to think about ways of targeting this epidemic of social isolation that we are seeing.
What is it?
I thought of an answer to the peptide question.
Okay.
Sorry, I thought of an answer to the peptide question. obviously GLP-1 peptides or like Ozempic, Wegovy, Mounjaro, whatever. Those are super popular right now. A lot of people are taking them. And anecdotally, there was like a lot of kind of reports that people just felt better, psychologically on these peptides. And so they ran a clinical trial, which is amazing. This is the kind of thing that we should be doing. The results were a little disappointing. There wasn't a clear preventative effect of Ozempic on Alzheimer's disease, but I think there's an enormous, room for improvement. This is an area that we should be doing more research on.
Absolutely, like multi-purposing drugs that are already approved and out there and widely distributed, and yeah, like you said, the interrelatedness of like obesity and diet. If you're improving that, it's going to have downstream psychological impacts. Even though that was like a, not a robust trial result, I think it's still intriguing.
Yeah, I mean, the peptide, I mean, that's fair. I mean, it's inside baseball. Like most people know it as GLP-1 or Ozempic, but yeah, the peptide. So, yeah. So there's actually another question for you, Leyla. Your work is convincing and exciting. Does your research only apply to people with ApoE4?
No, it applies to everyone. I think that there's a lot of evidence that these risk genes beyond ApoE are also converging on lipid metabolism. And these could be other risk genes, for example, lipid transporters, other lipid modifying enzymes. But I think another way to think about this is no matter what the kind of genetic profile is, obviously we learned today that Alzheimer's is such a multifaceted, heterogeneous disease, and that likely lipid accumulation could be resulting from many different pathways and sources. And yes, I would bet money that this kind of lipid pathology is present in Alzheimer's disease regardless of ApoE genotype, and that the research is hopefully relevant to a broader patient population.
So, Cara, in terms of studying amyloid plaques, the question is, I'm trying to understand the question to reframe it for you. It's essentially the disease progression. Can you describe to us exactly the disease progression that you would be seeing in a lab setting and how you would.
So when it comes to the tau pathology that we're modeling, we've done lots of different validations and measurements to kind of see whether it mimics what would be seen in an Alzheimer's disease post-mortem. And what we get probably at 3 to 4 weeks in a dish, maybe 3 months in a model, is quite close to end-stage Alzheimer's disease tau pathology. So when we want to look at the early stages, we're kind of looking at a week in our models in a dish, a week to 2 weeks, and maybe around a month in the mouse models. And then we think that that's probably the earliest tau events that we can see. So we're really compressing a big timeline into such a short space. But I think that makes it desirable to drug developers and pharmaceutical companies, which would typically avoid going after a target that they can't study quickly.
Okay. Jennifer, this is a question for you, or maybe Leyla, you can chime in. Did any patients receiving the Eli Lilly medication donanemab in the FDA clinical trial have an absolute reduction of amyloid plaques in their carotid artery? Either one.
Oh, that's a good question in terms of peripheral amyloid. I'm not familiar about whether that was measured or not in terms of carotid artery plaques, but given that it was introduced into the periphery, so had to cross the blood-brain barrier, which is usually the larger challenge, I would suspect that it could have an effect on the periphery. But I don't know if and maybe Leyla, you can weigh in on this, if the form of amyloid plaques in carotid vasculature is the same isoform as plaques in the brain, because just like tangles, there's different kind of stages of a plaque's lifespan in terms of microfibrils to protofibrils. So I don't know if that drug had an effect on the form of carotid amyloid plaques.
Do you want to add to that?
Just
Leyla?
that I completely agree. That I'm not familiar if there is any data on the carotid artery amyloid levels specifically with donanemab, but given that it's able to penetrate the blood-brain barrier, clear amyloid beta plaques from the brain very efficaciously, I would be surprised if it didn't have an effect. Yeah.
Jennifer, the question for you. What are your thoughts on supplements, especially collagen and creatine?
Oh, yes. Yeah. So there's, I mean, I'm just thinking about, social media, and we're talking about social media technology. And I was just thinking of my own experience. Like I've been looking at creatinine on social media, and now I'm getting fed almost every ad I get or story is about a form of creatinine, not yet collagen, but creatinine. But anyway, so not to digress, but yeah, so creatinine and collagen, a lot of buzz out there and interest. And definitely for the brain health space, again, not robust data that these two molecules alone, if we take them, they haven't been studied in a controlled fashion in terms of long-term outcomes. I think just for overall bodily systems, like given the benefit of creatine on muscle development and then collagen for skin elasticity and other bodily systems, I think most medical professionals will agree, like in moderation, if you don't have any other medical contraindication, but in terms of being like a robust, method to alone help brain health, I don't think it's quite yet at that level of evidence. That being said, I am taking a recommended amount of creatine each day with my coffee. It's easy enough, and I'm not like overly taking it because there's no evidence that taking more might be any better. So.
Yeah, there was the study that came out of the Brigham group, Joanne Manson's group, and Baker, where they showed for cognitive health the multivitamin.
Yes.
But I don't know the compounds in a multivitamin, if it has collagen or creatine, and and it didn't look at specifics. Yeah.
But that was a very interesting study about the multivitamins. That's something also easy enough to implement. Yeah.
And I think the results were like the very first year they saw results, and it stayed for 3 years with a daily multivitamin. Yeah. It was a clinical trial. Ganga, I believe this question is for you. Do you know if social and dietary factors are working through epigenetic pathways or some other mechanisms?
Both. Yes. So there are several of these aging physiological systems that are impacted by both the social environment and the diet, which is also impacted by the social environment. But yes, these epigenetic mechanisms, which are the ways that our gene expression are influenced by the environment. So, and it happens through, for example, methyl tags that change the way those genes function. And those epigenetic mechanisms are very sensitive to things like diet. So we know that as was mentioned, that the MIND diet, for example, or the DASH diet, which is targeting hypertension, are specifically implemented to reduce inflammatory pathways, for example, which are very sensitive to epigenetic mechanisms. And through those pathways and the social environment is acting, right? The social environment can be its own form of stress. So literally, I experience stress and it influences a cascade of physiological processes that act through these epigenetic mechanisms, really. And there is some evidence that shows that those mechanisms persist across generations, where once these genes are turned on, you see in offspring some of those residual effects. But certainly there are other ways that the social environment is acting to influence health behaviors. So physical activity, diet, cognitive engagement, et cetera. These are all ways that the social environment, which can act directly on stress, but also indirectly through shaping our health behaviors and our access to care, can act along these epigenetic pathways. And it's really such a fascinating body of work now, and it really opens up the door for us to understand kind of how aging is shaped by the social environment and the complexities around it. I think that that is really, again, loving the framing of challenging and opportunities, because when something is complicated, it really creates challenges, but it creates these opportunities now where we can move away from the focus on, are these just genetic differences, to understanding, are these epigenetic mechanisms that are shaped by the environment? So really great question.
Yes. Cara, accelerating tau clearance could be a key future treatment strategy in Alzheimer's disease. Do you see any associated challenges with this?
So I think the biggest challenge first is seeing the way that the brain cells do actually boost tau clearance. So we're working on understanding that. There's probably 4 to 5 key ways that brain cells do clear junk. So we want to see which one is probably the best one to boost. But I think possibly one of the biggest challenges will be that in the aging brain, we know a lot of these processes slow down anyway, even without any dementia. And I think it's then working out how can we actually boost that so that it can kind of deal with this extra load of protein clumps.
Thank you for that. And this is a question to the whole panel here. Can the presenters discuss the impact of the glymphatic system on ADRD and what behaviors people can do to stimulate the glymphatic system more?
I can start, definitely. So the glymphatic system is the brain's kind of ability to clean itself by pushing CSF, this fluid that kind of covers the brain, and using that to like take up all the metabolic waste produced by cells and flushing it out. And this process usually happens during sleep. So I would say, first off, getting a good night of sleep is super important, if possible. I'll pass the mic.
Oh, darn, that's what I was gonna. I was gonna say lymphatics and sleep, but it's actually, yes, along these lines, it's been shown through clinical research using PET that tau clearance occurs, a lot of it occurs in humans during sleep. And then, of course, we've known for multiple conditions, like sleep is protective for brain, for cardiovascular health, for mental health. So it's definitely one of those upstream modifiable factors, probably operating through the glymphatic system. I don't know of any other, myself, I'll pass this on to the next.
Yeah, well, I'm going to hop on the sleep train and say that people who are more stressed don't sleep as well. So one of the ways for certainly enabling these processes through sleep is to deal with your stress, right? And understanding what causes you to respond to certain stressors in a certain way and observing that and identifying ways for you to potentially deal with your stress in a different way. And that's, of course, much easier said than done, but certainly I think the more we know how stress impacts us, the more important it becomes for us to understand what we can do about it. And I think that will help you sleep better, which then will facilitate these processes. So, yeah.
Oh, no, just can I add one thing about sleep too? So if you think you are not sleeping optimally, it's definitely worth getting a sleep study to see if you can further optimize it. Like, even if it's not like, I'm not obviously snoring or I just don't feel rested when I wake up, it's definitely worth probing into why that is. And of course, there's multiple factors at play. But I think if you can, aim to address that and to identify it early, I think that's going to be a key upstream factor.
And the only quick thing for me to add, obviously sleep is important, but I know there is exciting research in the preclinical space where people are trying to come up with medicine approaches to boost lymphatic clearance. So that might be for people who cannot get appropriate sleep hygiene in the future.
Yeah.
Oh, you have something else to add?
No, I was going to say that would be us. That would be us.
Every small children, I don't get it.
Well, that was a great question, and you got everyone to chime in on that. I'm going to do my last. I have 49. Yeah, the time is running out, but lightning round. You're the future experts. Give me one thing that you would recommend to reduce someone's risk of dementia. It's not a question. I'm indulging in my own question. Go on. One thing.
Having friends and talking to them.
Jennifer.
Eating a rainbow-colored dietary plate.
Caring about other people.
I think one of the key things is get your eyes and ears tested because having corrected vision, corrected hearing is really important for reducing dementia risk.
And with that, let's thank this incredible panel. Well, please stand, and I'm going to do a quick wrap up. People who know me in this position know me for going a little long, but I'm going to go very short. And I'd like to thank you all, thank everyone on Zoom, and thank you to the rising stars. The future is bright. Thank you. Thank you for sharing your insights, your perspectives, your science, and your recommendations. It's hopeful. It's a devastating disease and all of the disease around it, but it's hopeful because they told us today that it is. So thank you and have a great evening.